We can talk about, how to do it better and fancier later, I guess. The basic gist of the format is going to be that I'm going to let them present their two papers and interrupt them as I feel necessary to clarify points or to better bring everyone to an understanding of the biological point they're trying to make with particular figures or with particular statements. And I have some background questions, and I have some things that I'd like to hit, but I don't know if we're going to hit those things. I'm not going to push this in any particular direction because I think the most important thing is to make it like a journal club. My nose is clean. To make it like a journal club, and that's what it should be. And it's a very special kind of journal club because we're privileged to have the authors of the paper that we are discussing with us, and that makes this journal club extra exciting. And so this is a really exciting opportunity for me as a biologist to talk to these people because I think this is, whether you like it or not, part of this world history that is forming around us. So I'm really excited about this. So I'm going to bring up their audio now, I think, and pretend like this is starting. I see a background picture there, so they're probably getting ready to start as well.
Yeah, Doctor.
Yep.
Let's start. Who's gonna be a first or starting you? Then we can make an okay.
Okay, very good.
唐教授，喂，
喂，
你再说两句。
喂，还有
多说几句，刚才怎么有回音？你再说几句。
现在呢？还有吗？
现在可以，严博士，你再说几句。
我现在应该可以吧？
好，
刚才说话回音很重。
好，现在没了啊。这你让那个人，好，我们开始啊，开始。5、4、3 ，1、2、3 ，start。
Hello everyone, welcome to today's Ludus show. Today we're going to have a special episode here, so we will have Dr. Li-Meng Yan, we will have Dr. Kang, and we will have our special guest today, Dr. Kui, and we will have an open discussion about the first and second Yan report, and it will be exciting. So first, it's my honor to introduce Dr. Kui. So Dr. Kui is a neuroscientist, and he did his PhD in Amsterdam, and now he is a staff scientist. Now he is a research faculty at the University of Pittsburgh since 2016. So today, we are going to have Dr. Kui ask about all the questions that he's interested in, or hear his comment about Dr. Yan's report. So first, to remind our audience, I will first briefly summarize the content of the first and second Yan report, and I will start the discussion. So as our audience may remember, the first and second Yan report describes how the virus, the COVID-19 virus comes from, and scientifically. So in the first Yan report, Dr. Yan discussed the scientific evidence that how this virus is made in a lab. So we know that this virus shares 89% general sequence similarity with the Zhoushan bat coronavirus, as reviewed by Dr. Yan first in January 19th in Ludus show. And specifically, its ORF8 protein shares 94% a similarity with the with the COVID-19, which is a poorly conserved protein, and most importantly, its E protein is 100% identical to the COVID-19. I mean, between COVID-19 and Zhoushan bat coronavirus, which means that COVID-19 has a clear template from a Zhoushan bat coronavirus. But on the other hand, the S protein, which is the part that very important to determine what receptor that the virus can bind to and what host range of the virus is poorly conserved. Is poorly conserved between the bat coronavirus and the COVID-19, and there are some key features such as RBM has been swamped, such as the restriction digestion site flanking RBM and the addition of the furin cleavage site. And the second part of the first EN report is basically a synthetic route of how this virus can be possibly made in the lab. Different steps versus swap RBM at furin cleavage site and make the PCR or DNA synthesis of other fragments, and then you ligate them together. Okay. And the second EN report basically is to show evidence to prove that all the other so-called virus that shares great similarity with COVID-19 are actually fabricated viruses, such as RATG13, such as the pangolin virus, such as the Yunnan coronavirus that has the furin cleavage site-like sequence. So, for example, RATG13, it has the problem of a fabricated sequence, and the author lied about the origin of the sample and the synonymous and non-synonymous mutation with the ratio of this synonymous and non-synonymous mutation between RATG13 and the COVID-19 doesn't fit the normal election rule, and most importantly, Dr. Yan disproves the minor passage hypothesis, which is completely based on RATG13 or a virus that shares a part. That virus has a fragment that shares 100% sequence with the so-called RATG13. And of course, the pangolin virus and the Yunnan virus that has a furin cleavage site all have, similar problems, such as the synonymous and non-synonymous rate problem, and the data problem, and the sample origin problem. And then at the end, in the second Yan's report, Dr. Yang concluded that the COVID-19 is a unrestricted bioweapon based on the fact that it is lab engineered and how the virus is used. Okay, so that concludes the first Yan and second Yan's report. So first I would like to ask Dr. Kui my first question, how did you start to get interested in the origin of SARS-CoV-2? And could you share this with us first a little bit? Thank you.
Sure, no problem. Thank you very much for allowing me to join this discussion. The thing that I should let you in on is that I actually worked at Erasmus University in Rotterdam, the Netherlands, during 2012 to 2016. These were the 4 years that followed the 2011 paper, where in the same university on a different floor, they had used ferrets to enrich avian flu to make it airborne. And this created quite a controversy in the Netherlands at that time, and being a generally interested naturalist and biologist, I was aware that this was going on while I was living there. And so I have always been interested in gain-of-function research and the idea that we are taking viruses from nature and tweaking them not only to understand them, but to understand how their function works by increasing it and increasing their virulence, or increasing their ability to transmit via airborne, or increasing their ability to replicate without mutation. All of these things we've studied before. So for me, when this whole story started in, let's say, December and January, and the WHO was saying things like that there was no evidence for airborne transmission, and our government was basically telling us we didn't have much to worry about. I was already aware that this is possibly not the case. That that the history of this is that since SARS broke out in 2003, many governments have been investing a lot of money in figuring out coronavirus biology, because coronavirus biology is interesting from the perspective that we have a number of endemic coronaviruses that could potentially be useful if we understood their biology. And coronaviruses in general are so abundant in nature that they are just an overwhelmingly large biological sort of section of biology that we don't know very much about. And then I saw a blog called Harvard to the Big House, and that was a blog which was written basically about the history of gain-of-function research and why this was likely also a gain-of-function biological sort of creation, and making the argument that dismissing this idea was foolish. And I don't remember when it is that Dr. Kang's blog came out, but it was after Harvard to the Big House had got me on this, and at the same time I was doing journal club on my bike. So I just made a journal club on my bike about this blog, and later on I saw Dr. Kang's NerdHasPower blog, and I did the same thing with that, and this was the first. Admittedly, it's the first real data that came out that didn't just sort of hand wave around the idea that we found this virus and we know exactly where it came from and don't worry. And I think at the time that your blog came out, it was still not really clear that the market story was complete hogwash, and it wasn't really clear that the pangolin viral sequences were under question. And now, as this narrative has progressed, we've gone from a very simple story in February to an addendum in Nature, which corrects this and admits that they found these viruses in 2012 and that there were other ones, and none of them are on GeneBank, and you're aware of all of this. So that's how I got there. And it's been a long time coming, I think, where every time we open this story again, we come back to the NerdHasPower blog, where these sort of incongruencies with the RATG13, let's say, consensus sequence, and their story about where it came from and their interpretation of what it means. And I think you guys, your paper is really focusing on this, of course, but more importantly, it came out at a time when most of virology, which is allowed on television, most of the virologists allowed on television were absolutely, positively, 100% dismissing the possibility that this could come from a laboratory. It was absurd.
Yeah. I think it's just definitely crazy that nobody even go into the details about all the genetic evidence and all the evidence and all the fact about the virus. Nobody provided evidence about the genome analysis, and then just simply claim that all from nature, and it's impossible that it is a lab-created virus. So that's why we are here today to present all the detailed evidence and have an open discussion for all of our audience to understand how this virus is originated. Now I would like to give my microphone to Dr. Li-Meng Yan, and maybe we can start with Dr. Yan. Introducing into more details about the two Yan report, and then Dr. Kui, you may ask questions and comments anytime as Dr. Yan speaks, and we will have this like an open discussion. Okay.
Very good.
So, Dr. Yan.
Hi, Dr. Gwen. Good morning, everyone. Good morning, Dr. Kui. Very happy to meet you.
Very happy to meet you.
Yeah, and I'm happy that we can share the chance to discuss the Yin report and also the COVID-19 things together in a live way to our audience. Thank you.
Yes.
And based on your description, so I just want to address some issues that maybe not only you have such questions. I have heard it for many times from many people when I met in US that they may say, so yeah, why you are the only one who just tell the people now in the media that this is the lab origin, and seems the other scientists are all holding another opinion that it comes from nature. And the top scientists also have that idea. The top organizations like the WHO and the NIH, CDC, the authorities also show these opinions to people. So how could I prove that I'm holding the right things or telling the fact? So I want to tell people that I don't want to prove that whether I'm right or wrong. I just show you the facts. So as I said, you see there are two reports. You have read it and you see the evidence here can make you understand better about the details about the lab origin, right? And also you asked that why before February, when you have the same idea that it seems not a natural virus, not a common virus, because you have the experience when you are a neighbor in Netherlands as a PhD student. You just understand some of the virology study and you hold this question, why there is no one else hold such question or tell it in public or discuss it even. Yes, so that is the thing I really want to clarify that. Dr. Kang, he write that blog actually based on my renovation from mid-January and also based on my very brief introduction on Genius I published on 25th January about the comparison between the SARS-CoV-2 virus isolated from Wuhan and Zhoushan bat coronavirus DC45-DXC21 and also known virus in the gene bank. So the result shows clearly that E protein identical similarity between the Zhoushan bat coronavirus and also the SARS-CoV-2 is the character that it cannot come from nature.
And I think this, just to clarify, to make sure people understood what you were saying there, this is one of the points in the original paper that the E protein, and correct me if I'm wrong, that the E protein is a protein that in general mutates quite rapidly and regularly as a virus moves through a population. So even if viruses are related to one another in a sort of hierarchical tree, we wouldn't expect their E proteins to be identical. Is that correct?
Exactly. Even among the human population, you see, at that time, only early February, there are not many human clinical samples isolate the SARS-CoV-2 virus, but there are already mutants happen in E protein among such virus samples.
Yes, so that is a really crucial observation. It's one of those things that they don't really mention when they mention this paper, and I think it is one of the biggest things in the paper.
Yes, actually, that's the thing. You never see those scientists who try to rebut my report. They never face to this problem because they know this is a fundamental evidence. And as I said, I have discussed with some experts in U.S. government, and they know it when they see this evidence. It's clearly shown cannot happen, especially cross-species, cross-human population, still keep such perfect 100% identical. And also, that is a very good example that in my second report, the Chinese scientists who list those novel zoonotic viruses, including the RETG13, pangolin virus, to RMYN, all these things, they share the perfect 100% identical E protein with SARS-CoV-2. It also tells people this virus cannot be real. And the only reason they share the 100% identical E protein is because there is no space for those scientists to change them, become the become the intermediate host style mutant. So is that clear?
Yes, definitely, it is clear. And
So
they wouldn't have also been, for example, could they have used the E protein as a way of also? I know that we'll talk about that later. That they use restriction sites to keep track of whether or not they kept the furin cleavage site in the spike protein or not as they grew the virus. But could they have also used this E protein as a marker? Perhaps is that possibly one thing they would? Or is that just a silly idea? I'm just trying to think of why they wouldn't have made. Couldn't they have pulled an E protein from somewhere else and avoided this issue or not?
That is a good question. So if you go back to check the quick diagnostic kit developed in China in January, there are several versions. Some combine two types of proteins from the SARS-CoV-2 to do the check. Some combine three. But the very common one is the one used ORF1AB, which is including the RDRT genome and also E protein together as an identical diagnostic kit to do the quick kit for the clinical patient. So those E proteins.
In your opinion, if they use those three proteins, that's a legitimate test then?
Sorry, could you repeat?
Yes. So if they use those three proteins as you described, then it would be a legitimate test for SARS-CoV-2. That's correct? Legitimate, like it's a good test then. It would work.
It's just a quick one. So as we know, always the golden standard is to do the PCR to see the genome. But compared to the time issue, it's a clinical one. So the quick one is very important. That's why it's asked for twice identical, twice diagnosed in the hospital. And actually in Hong Kong and maybe other countries, they use the PCR for the whole genome checking or the quick PCR. Better than this diagnostic kit. We can say that the Chinese case quality is poor, that only 20 to 30 or maximum 50% accuracy that is admitted by the China government. But still, including the ORF1AB and the E protein together, it's not the reason it's not accuracy. The accuracy is up to their technique, but that means ORF1AB plus A protein somehow can tell you that the character of this SARS-CoV-2. So these are two very important issues here. And also, what I want to tell is, So based on your question, there is another thing that why no one asked that. Actually, before I came to US, I already spent 3 months from mid-January. Before that, I tried to let people, the expert in WHO, get alert, but they refused to have response, as I mentioned. After that, I turned to Luda's media, delivered the message out in Chinese. We know there will be not only Chinese people listening to this video because this broadcast is very important, as you know now. My idea at that time is just to let more and more people understand. Even Chinese people, they can translate into English and let more English-speaking people to understand this information and go back like the scientists get together, pushed with the government to ask Chinese Communist Party open the discussion for the world and hold it in control as soon as possible. Somehow it failed, but you see, the alarm made Chinese Communist Party government very sensitive. So that's why they get response quickly after hours. Xi Jinping submit her RTD 13 paper the next day, and China government announced human-to-human transmission the next day, and then lockdown Wuhan after 3 days. But you should know that the Chinese Communist Party is not the government, care about people. And they have their plan to use this bioweapon for a long time. That's why after that, the virus comes from China to all over the world. And also, this is not just some accident that government are in a hurry to cover up or in a hurry to just organize something. You can see how the scientists' words respond from Lancet to Nature, to WHO, to those virologists, top virologists who you can list the name, right, a handful. So these people, their response is quite quick. Definitely not like you just heard there is an accident. You need to take time to think about how to hold it in control, how to help the government, how to save people. It's not in that way. You see, in Lancet, the release the email actually show people, even back in end of January, before WHO announced there will be an outbreak. When they still tell people that no point to get prevention, this is just like flu, they start to make the draft to praise Chinese Communist Party about their anti-COVID-19 contribution and also convince all the people this is a nature one and never mention lab origin. So after that, I was working in my lab, and I spent 3 months to collecting a lot of clinical data and a lot of information from China, which really helped a lot in this study, including the hamster paper and also Lancet paper about the viral load, and later antibody titer paper. They removed my name because I ran to US. And when I arrived to US, I around that time, end of April, I really realized that there is some information block. It's like US people have a firewall, you know? So what we have shared among our audience from January to April, you can see Dr. Kang write his blog in February. Dr. Guan also realized it before the February because they listened to the broadcast. They did independent investigate by themselves and convinced by that to realize the problem. But most of announcement people, they didn't know this. And the scientific world are very powerful. They can influence the public, they can influence the government. And also the scientists like you, although you hold a lot of doubts, but no one can help you, and because you are not expert in virology, so you need to get the help from those experts who just mislead you to the other direction.
No, I agree, and I think that this is
reason you need to figure out by yourself and give you a lot of difficulties, right?
Yes, I think one of the issues here, and I didn't want to go here so early, but I want to ask you specific questions about the paper as well, but the papers as well. But for me, I like the fact that you're most interested in getting the concept out there that all around the world we are doing gain-of-function research, and so this leak could potentially also happen in North Carolina in America. It could happen in Texas, and it could happen anywhere where these labs that do this research exist. And so I'm not suggesting to you that I'm just saying that in general, one of the issues has always been for me is to get this idea out there in the public that this research occurs, and that if it's done without the proper controls or without the proper, let's say, good motivation, we can very quickly have, even if this was not, and I still want to clarify, and I respect your opinion, of course, as a virologist, but one of the most inflammatory parts of your work is that you call it a bioweapon, and you accuse the Chinese government of making it with the intention of using it. And that is, of course, one of the reasons why people push back on it so much, because they don't want to believe that portion of the story. Yes. But the other side of this is that you could come up with a million different reasons in a grant application to do the exact same experiments, which would result in these types of viruses existing where they didn't exist before. And all of those justifications would be on Peter Daszak's resume as why he's doing gain-of-function viral research, and yet still could result in this virus being accidentally or intentionally released. So for me, that's the part that excites me the most about your statements in the beginning, that you're actually making that the pretense that it's a considering this. You have to consider this possibility before you can think about it rationally, and most people have not even considered the possibility. Go ahead, sorry.
No, thank you for your explanation. Actually, you have mentioned two parts. One is about gain of function. The other is about bioweapon. Let's first talk about gain of function. China is definitely not the only country which conducts gain of function experiment. Actually, back to 2015, that Dr. Shi Zhenli has learned her technique about swapping the RBM, receptor binding motif or receptor binding domain, in North Carolina with Dr. Barrick. And that is the thing make a lot of controversial. And then later, this kind of experiment, not only in that coronavirus lab, also like in highly pathogen avian influenza labs in US and other labs get suppressed for a while. But you have to know that always scientists, you are doing the neurology and we're doing the virology. Part of the reason they do research is because of
curiosity, yes.
So So the point is whether they can do or not is you have to get a very good supervision, that kind of structure. So for example, you do something like, for example, you do the things in P3 or in P4, that is definitely some dangerous experiment, even if it's not gain of function. It's lethal. It can be high contagious. But the thing is you have to check the lab, maintain the facilities, and train the staff, and with your responsibility, also with the laws, with the discipline, very strict ones. So like in my lab in Hong Kong, the top coronavirus lab, we have done the very strict background checking in the police station, and government know us very well. We get those through the two part of the examination and train hand by hand by the experienced staff in our lab, and also we maintain the equipment very well. Every day, morning, night, we check it, and we have the WHO or other people go to do the surveillance every year to check whether it's qualified. All these things. So whatever you can do or you cannot do, that is under the policy of the government and also up to the control from the government, right? So this is like in US, I know that there are some other labs still will do something. Maybe you call it gain of function, or you said it's just a similar experiment. But I can say that in US, the government surveillance still is much better than in China, than in mainland, because you have your regulations. But in China, as I listed in my report, for decades back from SARS time in 2003 and even longer, our government encouraged such dangerous experiments. And also, I'm from China. I studied there, got my PhD, MD, and also have a lot of friends working there still. I know how these people manipulate their experiment. I mean, basically, you can refer to 2018 that Dr. Hutchinson's HIV CRISPR editing baby experiment. So this kind of dangerous things are encouraged by government, even using human trials. Even use very immature experiment. So in this level, gain of function experiment, whether it's so dangerous, whether we need to worry about, you can see the clear difference between China, between Chinese Communist Party government and the Western countries. So it's more risk, when China is doing that, right? And the other thing is we are talking about bioweapon. I tell people this is an unrestricted bioweapon. But as I mentioned clearly in the second report, this is not my concept of bioweapon. Bioweapon, this concept is based on the top bioweapon expert in China military of medical and science academy. Professor Fu Yang, who has conducted the Iraq bioweapon examination program with international experts before, he told it in public that the standard with Chinese Communist Party, the Chinese Communist Party, won't develop as bioweapon. The first thing is it can cause people either hurt or death. We see SARS-CoV-2 virus won't make people die like Ebola because that is not good transmissional model. It can be located in a small location very quickly because people realize something wrong here. But it causes a lot of injury. People get sick. People who are have underlying disease suffer more, and the kid may have the chronic influence. And people also have like the when they get even recover, so-called recover, there are even 10% to 30% people, they may suffer from the neurologic something can that based on the recent data, right? So we still don't know what exactly happened after the infection of COVID-19 to the whole population. That is a very, very big problem. And the second thing is that we